This presentation on an overview of linkage maps is sponsored by the Tools for Genomics-Assisted Breeding and Polyploids Training Workshop and funded by NIFA, the National Institute of Food and Agriculture within the USDA Specialty Crop Research Initiative, and was held on January 14, 2021.
Welcome to the linkage and phasing in polyploids. So this is an overview for the next 3 sections. So here I will try to explain the ideas behind linkage and phasing in polyploids. And when I do this, I always start explaining a little bit about diploids, and then we jump to polyploids. So if the diploid part is too basic, just bear with me, and then we go to the next level, okay? Yeah, so let's start with the basic concept of linkage. So linkage is something that is well understood for diploids, and is a very self-explanatory concept. When you have two things linked, it means they are close together, right? So here we have 3 examples of 2 markers in diploids. And here we have 2, let's say, genes or markers. We have marker A and B. And in this first case, they are linked, they are together. And in the second and third case, they are far away. In the third case, they are in different chromosomes. So we say they are not linked, okay? But the most important thing here about linkage is not where they are in the chromosome, but the consequence of this fact. So if they are close in the chromosome, okay, they are more likely to be inherited together. So this is the main idea, and that's why this concept is so important, right? And the closer the markers are, the lower the probability of a crossing over event occur between them, and consequently they are more likely to be inherited together, right? So this is the basic concept of linkage, and this applies to diploids, tetraploids, exaploids, and so on. And how can we measure how linked 2 markers are in the context of genetic linkage mapping? And yeah, so we use something called recombination frequency or recombination fraction in segregating populations. Here, I'm going to explain why we do, how we do this in biparental populations, right? But this is a concept that can be applied to families, and but here we will stick to the biparental populations, okay? And genetic linkage is a concept applied to at least 2 loci. So you need 2 positions in the genome to see how they are linked, right? And the recombination fraction is actually a probability. It's a probability that an odd number of crossovers occur between 2 markers. It ranges from 0 to 0.5. This is considering no double reduction, okay? Because this can be a higher number when you have double reduction. And during this presentation, I will not consider double reduction, because it's another layer of complexity to a subject that is already very complex. But we can discuss this. Since recombination fraction is a probability, and we are oh, there's someone that needs to mute the mic. Okay, so since it is a probability and we are talking about distances, we need something to transform these probabilities into distances, right? And we call this mapping functions. There are several mapping functions. The most common ones are the Morgan mapping function, Haldane, and Konsumbi, and there are other mapping functions that actually use some properties to transform these probabilities into distances. And when you compute the recombination frequency between the pairs of all markers you have in hands for your genome, you can group these markers into something we call linkage groups, which represents basically the chromosomes of the species. And within each linkage group, we usually order these markers, and or we can order using a lot of different algorithms. Today, we are going to present the MDS algorithm, which is a very, very nice algorithm for hundreds or thousands of markers. But you also can use the genomic order. But this is for the linkage. But first, let us address the behavior of just one locus, a single locus. This is not loci, it's locus. Just one single locus when they go from the father to the siblings, okay? And this is, yeah. So here we have the first thing we need to understand is that in polyploids, we have the meiotic process is very unique, and you can have several different possible configurations in this meiotic process. Here we have 3 examples for a tetraploid. We have 2 bivalents where 2 chromosomes they pair 2 by 2. Here we have a tetravalent where they are pair all together, forming crosses. And this is, here you have 1 univalent and a trivalent pairing. But for this presentation, let's stick with this first case here. And let's try to imagine the gametes that we have when we have this first case, okay? So basically, in a tetraploid, in this example, we have 4 possible homologs. And we have A1, A2, A3, A4. If you think in a balanced gamete with the half of the level, we can have 4 choose 2 possible gametes. So in this case, it's 1, 2, 1, 3, 1, 4, 2, 3, 2, 4, 3, 4, with no double reduction again. If you are considering double reduction, you would have 1, 1, 2, 2, 3, 3, 4, 4. But in this case, just let's stick with this more simple example, okay? But usually, to assess this information, instead of multi-allelic markers, what we have so far is are biallelic markers. And biallelic markers, as Roland said yesterday, they give you the dosage of that specific position. So in this case, A1 and A2 are represented to big A and big A, and A3 and A4 are represented to small a, small a. So you have a 2-dose or a duplex marker in this case. So let's change the 1s and 2s here by big A's and the 3s and 4s by small a's. And you have this sort of configuration here, right? You have big A, small a, 4 times, and small a, small a. And then you have the segregation for this gamete as 1 to 4 to 1. So this is a segregation with no double reduction for the tetraploid case in a biallelic marker. So this is a table with all possible gametes for several ploid levels. So for exaploid, for tetraploid, as I said, we have 6 for the gametes. And when you combine gametes from both parents, we have 36 possible genotypes in your population, in your biparental population. For exaploids, we have 6 choose 3, and we have 20 possible gametes. And if you combine 2, we have 400, and so on, up to 16. So you can see that this number grows explosively. It grows very, very fast, right? And this is a problem. And this is, I think this is the main problem in polyploid linkage analysis. So just to explain this better, let's start with this diploid concept here. So imagine I have 1, 2, 3, 4. Now I see that's a bit small, but it's 1, 2, 3, 4 here. And then we can have gametes 1, 2, or gametes 3 or 4 here. And when you combine, we have 4 possible genotypes in a diploid, right? And they will segregate 1, 2, 1, 2, 1, 2, 1. If you are just using biallelic markers now, we do the same thing that we did with the tetraploid. We have our famous 1 to 2 to 1 in a diploid, right? But now, let's imagine the same phenomenon with tetraploids. So in tetraploids, as I said, we have 6 possible gametes from one parent, 6 possible gametes from the other parent, and we have 36 possible genotypes. But since we assess this with biallelic markers, this information is somewhat hidden by this lack of information, this lack of information of the biallelic marker, because here you can't distinguish between big A, and small a. And this is just an example with a 2 dose. And in this case, we have this type of segregation, 1 to 8 to 18 to 8 to 1. And again, this is with no double reduction and assuming that we have random pairing. We are going through this in a bit. And for exaploids, we have this. We have 6, 6, and the reasoning is exactly the same, okay? And as you can see here, it's just the way you change your A, 1, 2, 3, 4, 5, 6, and so on by big A's or small a's, and you have different combinations in this matrix here. And for this case, 2 triplex markers in exaploid, we have the segregation 1 to 18 to 99 to 164, 99, 18, and 1. Okay. Oh yeah, just let me say something. All these images, they are available in my website. If you want to use them to teach in your classes, they are all available, and you can make changes, and they are all in my GitHub account. Okay. Okay, so now let's jump. Let's change from one position, one locus, to 2 locus. I will explain a little bit about recombination fractions in diploids. And this is a very common example. So we have 2 lines here. As you can see, we have big A, big A, big A, big B, big B, small a, small a, small b, small b. We have F1, which is a heterozygote, right? And then let's back cross. This is the first genetic design we learn when we are learning linkage. So when you have the back cross here, this individual, it doesn't matter if there are crossing overs between the 2 loci here, because they are non-informative. But in this case, we can detect the crossing overs, right? So we have recombinant events and no recombinant events. In this case, they duplicated and they exchange pieces of the chromosomes. And it will generate gametes, non-recombinant gametes, recombinant gametes, and here we just have non-informative gametes, right? And when you combine this, you have sets of recombinant genotypes and non-recombinant genotypes. And to estimate the recombination frequency or fraction, you can actually just divide the recombinant gametes here by the total gametes. This is very trivial and very simple. But also it's very limited when you try to expand this idea to more complex cases. And the way we usually do this when we are trying to expand this idea is to use a likelihood concept. Okay, so we have here our heterozygous individual, and we have our recombinant gametes and non-recombinant gametes. And what is important in this particular case is to have the probabilities of each one of these events here. So we need to have the probability of observing A, big B, given a certain recombination fraction, and so on for the other cases, right? And the way we usually do this is to put this elements in a 2x2 matrix, and here are the probabilities. So we have the recombinants. Is the probability of observing a recombinant is R, right? Since we have 2 cases of recombination here, is R divided by 2, right? And the non-recombinant case is non-recombinant, 1 minus R divided by 2. So here we have all possible outcomes for our back-cross design, right? And what we can do here is to observe our back-cross population, and for each one of these, the individuals in the back-cross population, you choose one of these outcomes here, you get the probability, and you multiply this probability given a recombination fraction, right? And when you have this, this is the likelihood function for the back-cross, right? And in this case, we have n individuals, so we need to multiply over all individuals. And then if we maximize this function, which means what is the value that R assumes that maximizes the function, that provides the maximum value of this function? So this is this argmax operator here is doing. So what's the value that maximizes this function? When you have this, we have the famous maximum likelihood estimator of R, which is R hat. So to exemplify the situation, let's do a toy example, right? So imagine I have for marker A and B, I have my SNPs, they are called, and we have so these SNPs here in the pink ellipses here, they are big A, small a, and here we have small a and small a, and same for B, right? So I observe individual one, and our individual one is, so just ignore the second letter here because it's not informative, right? So let's just acknowledge the first letter here. So for my first individual, I have big A, big B, big A, big B, and so on. For the third, I have a recombination here, and so on. So we have two recombinant events here, two recombinant individuals, 6 and 3. And of course, if I divide 2 by 10, which is the total number, is our recombination fraction is 0.2. Another way to do this is to, instead of counting these numbers here, is to associate to each one we saw in the previous matrix and assemble a likelihood function, which is in this case is R divided by 2 to 2, 1 minus R divided by 2 to 8, because we have 8 non-recombinant events and 2 recombinant individuals. And what we can do in this case is maximize this function. There are several ways to maximize a likelihood function. There are EM algorithms, Newton half-sum, Brent methods, but here it's a very naive, still very efficient way to do this. So we just try several values from 0 to 1 in this case, and then we maximize, we use the maximum point of this function. And then when you see this, the maximum likelihood estimator here is actually the same we obtained here. And you might think, oh, this is a lot of trouble just to get the same value. But the thing here is not the approach, is the concept. This is a much more broad concept. And if you want to understand how to do this in a deployed, I have in my GitHub, I have something to explain how to do this in real situations with C++ and R. And everything is there explained. And also, there's actually something to build a package to do this. Yeah, so this is for the diploids, right? So let's talk about the polyploids. So the first thing we need to understand here for polyploids is that the meiotic process is different. And here, as I said, let's assume that we have just bivalents or at least small segments of the chromosome, right? And they will find each other during the meiosis. Let's say we have this first configuration. So this is an exaploid, okay? We have 6 possible homologs. And homolog 1 found homolog 2, 2 paired with 5, 3 with 4. This is one possible configuration. In this case, I'm calling this configuration psi 1. Here we have psi 2, where 2 is pairing with 4 and 3 with 5. and so on. We can have several. Actually, in this case, we have 15 possible configurations, right? For any ploid level P, this is the number of configurations. For tetraploid, this number is 3. For exaploids is 15. For octaploids is 100 and something. It's also a very, very explosive number. But looking at this formation here, we see some similarities with the diploid example, right? So my question is, can we model each one of these bivalents as a diploid? And the answer is yes, we can. We can use the same matrix, but we need to use a trick here called Kronecker product. A Kronecker product is just an operator that makes combinations, right? It's applied to matrices, but it's for combinatorics. So what it does is combine the first element of this matrix with the first element of this matrix with the first element of this matrix, and then the first, second, first, third, for all possible combinations. And in this case, when you combine all possible elements here, you have a matrix 8 by 8, telling you the joint probability of observing a certain gamete in position A and a certain gamete in position B. But this is given a specific bivalent configuration, okay? And interesting enough, we have this closed form for these elements here. So you can have any ploid level, actually. You can have from 1 to 8, 10, 12, and this will work, okay? And we have this closed form. And in here we have the recombination fraction, the ploid level, and the number of recombinant events we observed between the two gametes, the two loci. And as I said to you, this is just for one possible configuration, right? And we have 8 possible, an 8 by 8 matrix, meaning that we have just 8 possible genotypes in that configuration. But, and I also said to you that for a exaploid, we have 20 possible genotypes. Where are the other genotypes? They are not possible if you are just using one configuration. They will be present in the other configurations. And in this case, I have all possible configurations for an exaploid, 15 possible configurations. And I did the same thing I did in the previous slide for each one of the possible, for each one of the configurations, right? And to obtain the final matrix, which will be a similar to that small matrix you observed for diploids, but here will be a 20 by 20 matrix, we needed to sum over all possible configurations using the probability of each one of these configurations. So here, since we are assuming that this is an auto-polyploid, each one of these configurations has the same probability, right? But you also can use different probabilities, and you have different matrices here to use in the estimation of the recombination fraction if you have different preferential pairing profiles. And then, of course, if you sum over all possible matrices here, you have your the probability of observing 2 genotypes in an exaploid or in an octaploid or in any ploid level. And this is for one parent. If you want to do this for both parents, again, we use our trick, the Kronecker product, and then we just multiply this blue matrix here by another blue matrix. So if you have an exaploid with a tetraploid, you can multiply these matrices with different dimensions as well. And all the theory behind this is explained in this article we published 2 years ago. And okay, so since the exaploid example is too complex to show here, as a toy example, just let's imagine the same idea that we had for the diploid. Let's imagine here in a tetraploid context, right? So we have our 4 possible homologs. We have A1, A2, 3, 4, 1, 2, 3, 4 for B as well. And then here we have a non-informative individual, okay? So 2 nulliplex cases, and here a completely informative individual. And I did this just to consider one gamete, right? So it's easier. So in this case, using that equation, we can have this matrix here, and we can do the same thing we did for the diploids. You have your population, you observed your multi-allelic cases, and then you can maximize your likelihood function and obtain your recombination fraction as well, right? So this is very similar to the concept we applied in the diploid. So in this case, and that's why it's important to use the likelihood instead of just using the number of recombinants divided by the total, because this is it's much more easy to extend from that situation to this one. But in reality, we know that we don't have this type of markers here, right? This is very difficult to find one of these markers. And since we are using dosages, it's impossible. And how can we translate this to a dosage context? It's actually very simple. And we did the same and we can do the same thing we did when we are just using one loci. We just change the 1s, 2s, 3s, and 4s in this case by big A's and small a's. And there's a special way to concatenate these columns and rows here. But since you just have 3 classes here and 2 classes in the second case, because we just have 1 simplex marker here and 1 duplex marker here, a matrix 3 by 2 is sufficient to have the likelihood for this case. And again, you use this and you have your likelihood function. You can maximize this likelihood function, and you have the recombination frequency, right? The N1, N2, and N3 is just the numbers the numbers of the genotypes we observed in the biparental population. But in this case, we have a duplex simplex marker in association, meaning that the 2 allelic variants here are in the same homolog. But we don't know this, right? What we know is just we have 2 doses and a single dose. But we can have the likelihood function for the second case as well. And as you can see here, this is the first case. This is the second case. There's a subtle but very important change in the likelihood function. And to see which one is the best one, you just compare the likelihoods of these 2 situations. And when you compare these likelihoods, you can choose the most likely configuration, and you can assume that is the best configuration for your analysis. And of course, you need to do this for all possible markers, at least within a linkage group. And if you have these markers, these recombination frequencies between all pairs of markers, so meaning pairwise in your linkage group, what you can do, or in your genome, if you have in your genome, you can assemble using clusterization methods to assemble linkage groups, and then you can convert this to distances. You can use algorithms to order, and I'm pretty sure Peter and I we will explain some of these algorithms. And once you have the order, and you have this rationale to choose what is the best phase, you can figure out what is the best phase just using these 2-point estimations. Okay, and you can extend it throughout the whole linkage group. This is just one example of an algorithm called MDS. We are going to use this today. And it's basically just a way to reduce many dimensions, preserving the observed distance between points by minimizing a loss function. And what this algorithm can do is just to organize this very unorganized matrix in a very smooth and beautiful matrix like this one. And this was proposed in this paper by Catherine Preedy and Christine Hackett. It's a very, very nice paper. I highly recommend to read. And as you can see here, this is the shape of an ordered matrix or an ordered linkage group, because the markers that are together have higher recombination frequencies, and the ones that are far away, they have lower recombination frequencies. And yeah, so this is the linkage part using 2-point analysis, right? But now let's talk a little bit about haplotyping in polyploids. So for the haplotyping polyploids, it's basically, I have this situation here. I have 7 markers with several dosages here, 2 and 3 and so on. And my question is, how can I position these variants in the homologs of my species, right? So this is an exaploid. I have these variants here, and how are disposed, how they are distributed in my homology group, and then consequently we can have the map for this, right? So to answer this question, I previously answered a part of this question, right? Because we have the 2-point information, and I'm pretty sure Peter will discuss this in Polymap R, how to use this information to choose the best homolog to place your variants. So this is the first part of this procedure. And here I'm going to introduce something that we call the multilocus linkage analysis. And this is kind of a complex topic. I will try to explain here, but the most important thing here is to understand that instead of using 2 markers, we are using multiple markers to obtain the recombination frequencies and the likelihood associated to that particular phase configuration, right? So in this case, we have something for the multilocus linkage analysis. We usually use something that we call hidden Markov models, right? And the hidden Markov models depend on something we call transition probabilities, which is basically a probability of observing a certain genotype given another genotype in a previous position, right? And for any ploid level with no double reduction and no preferential pairing, we have this formula here, okay? And this is very well explained, including double reduction in this paper here. It's a very nice paper, and for tetraploids, and this is our paper we published expanding for any ploid level. And just to show you the mechanics behind the Markov model, imagine we have this situation here, is very similar to the previous situation. And then I observed these individuals here. So these are completely informative individuals. And I have something like this. I have 6 possible gametes. I'm just ignoring this individual here because it's not informative. And then using the first individual here, I have this path from 1, 2 to 1, 3, as you can see here, from 1, 3 to 3, 4, and 3, 4, 3, 4. And I can use that probability that I show you in the previous slide to do this for all possible individuals in my population. And I can do the same trick of maximizing my likelihood and obtain a recombination fraction for the whole chain. So it's not anymore just one, it's no longer one specific value is a vector of recombination frequencies. But this is where the things get complex, because we don't observe the complete information in this case, and we need to use something that we call emission functions, which is the hidden part of the HMM or hidden Markov model. And in this case, instead of 1, 2, 3, and 4, I have my big A's, small a's, big b's, small b's, and so on. So instead of navigating through the points, I navigate through chunks of my chain. And I can do the same thing of maximizing this chain for my second individual when I use all individuals together. And in this case, for instance, if I'm not sure if it's, in this case, big C, small c, small c, small c, I can transit or I can visit all states in that position, but I visit with a stronger confidence in the states that I observed with a higher probability. But in this case, I represent something here that actually is a misclassification. And when you run the hidden Markov model, it fixes itself because it's a iterative process. And you can test also different linkage phases, right? But instead of just using one marker, we use two. So this is a comparing two different linkage phases. So here we have one possible linkage phase for the parents and another linkage phase. The resulting hidden Markov model will be different when you can use the likelihood to compare. And this is a simulation which tries to show you a real scenario, right? So if we have, so this is a cross between two tetraploids, and these are the haplotypes of the hexaploids, right? Here are my 4 homologs for parent 1, 4 homologs for parents 2, and this is one particular individual in the biparental population, okay? And this is a real hidden Markov model for this very small example. So as you can see, each one of these edges here, they are actually operations. They are trivial operations, but there's a lot of operations. And this is for a 36 state hidden Markov model. For exaploids, we have 400, and you can imagine the complex of this. And when you run your maximization procedure here, it does something like this. So you start with all possible connections, and when you run we are using an EM algorithm or a Bauman-Welch. It's a very specific EM algorithm for a hidden Markov model. You see that you have all possible configurations here based on your dosage, but when they talk to each other here, at the end, you can find the very specific haplotype for this particular individual here. And you can do this, and actually the procedure does this for every individuals in the population, okay? And this is very interesting because we can find the recombination point right here, because here we have all possible genotypes, and then they are the same genotypes in this position along the chromosome. Oops, recombination here, and then it's another genotype. This is just the way that I do in NetPoly. This is how I implemented. I use a two-step of 2 points, and after that I use the hidden Markov models. I will explain during the NetPoly section, but I am pretty sure that Peter and Xiaozi, they have different approaches, and this is just one of them, okay? This is just a resulting map using this approach. This is our sweet potato genetic map. There's some things, very interesting things here. We have our map for one of the linkage groups. Here we have the haplotypes, or actually we have the full haplotype for both parents. And here we have something called genomic information content. It's basically something that we use to, it tells you how much you can distinguish between homologs. but this is just the result of the map. And this is a very important thing. This is one of the last slides. So when you use the probabilistic approach to reconstruct the haplotypes, you have several noises in your data. And you need to take into account those noises because you can introduce false recombinant events there, right? Because you have a lot of noise in polyploids, especially in exaploids. And if you let your hidden Markov model adjust itself, if you give you a prior probability of being wrong, what the hidden Markov model does is this one. This is real data. This is a real tetraploid. We are going to analyze this. It just it irons out these bumps in the probabilities of the homologs that were inherited by a specific individual in the population, right? So here we have, so these are actually probability profiles. And what we are in each one of these breaks here mean that we had a recombination between homologs B and C and a recombination between A and A and D and so on. And this is an example of an exaploid with much more noise. When you let the hidden Markov model works in the exaploid, you have this. And this is very, very useful and very important to do to have a very good QTL analysis after this, because this is the result that you use for your QTL analysis, because you're linked these profiles to your phenotype. That's it. That's the references. And yeah, thank you. Yeah.
This presentation has been sponsored by the Tools for Genomics Assisted Breeding in Polyploids Research Project. For more information, you can email us at toolsforpolyploids@tamu.edu or visit our website at www.polyploids.org. This project is funded by NIFA within the USDA Specialty Crop Research Initiative funding program.